Okay. Hi, everyone. Thank you for coming to the review session. I know it's very anxious, but we're here to just help you polish up whatever you need for the last bit of your review. And today, there's not a lot of topics on this exam, so probably expecting more in-depth questions on the few topics that will be showing up on your exam. I don't know. I'm gonna put this joke in here. I don't really get it. Like Kendrick Lamar gene structure. I guess there we go. It is what it is. So let's talk about transcriptional control. Let's begin with prokaryotes, right? So sigma 70 is a transcription factor found primarily in prokaryotes. It is essentially the main transcription factor that is required by RNA polymerase. So what happens is before RNA polymerase can bind onto the DNA, sigma 70 must bind to the RNA polymerase. This creates the full hollow enzyme, right? And this, complex between sigma 70 and RNA polymerase is what actually binds to your promoter. And it is consensus sequence specific. It binds to the -35 and the -10 regions. Yeah. All right, moving on to operons. Operons are, again, mainly found in prokaryotes. They're a bundle deal, essentially. So you have a bunch of related genes all grouped together linearly on the DNA. They're all regulated by a single promoter, operator, blah, 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 blah. You get it. And we can regulate these operons with essentially activators, repressors, and a combination of like attenuation as well, as we saw with the trip operon. Yeah, each operon contains regulatory binding sites for the regulators, and they can either be inducible or repressible. In our class, we're mainly focused on repressible operons because the trip operon is a repressible one. The trip operon. So the trip operon is responsible for creating proteins that are related to the biosynthesis of tryptophan, right? And so you see these genes are coupled together and they're all regulated by something called the trip repressor. And when trip levels are low, the bacteria need to express the proteins that allow for the biosynthesis of tryptophan. Like if you don't have a lot of it, we need to create the proteins that allow us to create more. And so, when trip levels are low, there's not enough tryptophan to bind to your trip repressor. The trip repressor doesn't bind onto your promoter. And so transcription is allowed to occur. When trip levels are high, right, there's two different regulations. There's attenuation and the repressor. So when trip levels are high, not only are the repressors bound to your promoter, but also attenuation occurs because the ribosome does not stall on the leader sequence.
We'll get into like the details of each scenario in like a second.
So this is right when we're talking about the regulation of the trip operon, you need to think about spatially the difference between the transcription and the translation. So on your DNA segment, you contain not only just the genes for the trip operon, you also contain the leader sequence. The leader sequence is part of your mRNA, but it's regulated primarily through translation, right? So do you have another slide for this?
What?
Do you have a slide for attenuation?
Yeah.
Okay. We'll get there.
So basically, this is a breakdown of the first part, right? So at the base level, you have this is the entire gene. This is the genes that you're coding for. This is the promoter where the RNA polymerase binds. This is just a gene upstream that codes for the repressor itself, and this is the operator.
Nope.
Okay. This is the operator where the repressor binds, right? And at a fundamental level, this X kind of similar. Do you guys remember the lack of now? From 151. Yeah, for the most part. Basically, it acts.
Yeah, get closer to the mic.
It acts kind of similarly in that the TRPR gene codes a TRP repressor. If that TRP repressor is unbound from TRP, right, it is inactive. When it is bound to TRP, it is active, right? Because there is TRP in the environment, you don't need these genes to be expressed. So TRP binds to the repressor and attaches to the operator. If it's attached to the operator, it's physically blocking RNA polymerase from traveling down the length of the gene and transcribing those genes, so you don't get expression of the TRP operon. Does that make sense, everyone? Okay. If anyone has any questions, feel free to like raise your hand or interrupt. Yes.
Can you talk about how there are two levels of information at the requested?
Because attenuation
Yes.
can only occur when transcription first starts.
Yeah. Wait, what shouldn't? What? So we'll get to that in a second, but basically there is a scenario. If you think about what TRP is used for, right? Tryptophan's, I can't see shit. If tryptophan's, like tryptophan's an amino acid, right? So it's used to code for proteins. So the way that it does that is it associates with tRNA to bring it to the ribosome, right? The anticodon of that tRNA corresponds to TRP. So you're trying to code for TRP in an mRNA sequence. So even if you've run out of TRP, how do you run out of TRP? Right? Free TRP, what is that free TRP used for? It's used to make TRP tRNA, right? Each one of these little triangles is associated with a TRP tRNA, right? That has the correct anticodon. So you can have a whole bunch of TRP tRNA even when you don't have this, because you've used all of this to make that TRP tRNA. You see what I'm saying? Yeah. So basically what happens is you kind of have two like up down swings, right? And one lags slightly behind the other. And the one that's lagging is TRP tRNA. So if you have a lot of TRP, that's used to replenish your TRP tRNA, right? After that, and then you use up that TRP in replenishing your TRP tRNA. So then you run out of TRP, but you still have that TRP tRNA. Eventually that TRP tRNA gets used up. Now you don't have anything and you need to express this operon. Does that make sense? Yeah. There are basically three scenarios that can occur then. One is you have TRP, right? If you have TRP, does it matter whether or not you have TRP tRNA? And why? You shook your head no. Everyone else kind of looked at me. Exactly. So regardless of the level of TRP tRNA, it's going to stop right here, right? If it stops right here, then the reason for that is that TRP that you have can be used to make TRP tRNA. If that TRP is used to make TRP tRNA, then you have that situation where you have low TRP and high TRP tRNA. Eventually that TRP tRNA gets used up and you have the third situation, which is low TRP and low TRP tRNA. That's when you actually get expression. Does that make sense?
Can he repeat that?
Holy crap, we can hear you guys. Okay, I thought we couldn't hear you guys. Which part?
The three different scenarios, just to solidify.
In a half a second, like I have like slides for each of those.
Oh, okay, perfect. Thank you.
That's awesome. I'm sorry, that scared the shit out of me too. I'm sure it spooks some of you guys. I don't know how to
Sorry
lower
about
the
that.
volume though, so. Oh wait, no, we can. Wait, Isabel, can you say something again?
Hello.
Is that
Okay.
better? Okay.
That's
can
better.
all-
I didn't mean to scare anybody. Sorry about
Oh,
that.
That's fine.
You're good. We just I couldn't figure out how to work the volume. All right. So the next level, right? The first level was what we talked about here, which is like the TRP control, which is controlling whether or not RNA polymerase is bound and actively transcribed. Right. So that's step one. Step two is attenuation. Right. So keep in mind that this is an operon. Where do operons show up? Here's a question for you guys.
What type of cells, essentially?
for the people. You
Makes
said
sense.
that. Hold on. Operons generally show up in prokaryotes, right? Because eukaryotes have better ways of regulating. They don't really need to do this. If you think about how an operon works, it's basically a crap load of genes that are encoded for within one mRNA sequence, right? The reason for that is I have a whole bunch of DNA in each one of my cells because I'm a eukaryote. Prokaryotes only have like a tiny, tiny fraction of that, right? So they have to be really efficient in using their space, like in the genome. So they can't have every single gene have a whole promoter, a whole operator, a whole other enhancer, whatever. So they cut down on that promoter, enhancer, operator space by just lumping a bunch of genes together. So that's why you get operons a lot in prokaryotes. The reason the trip operon works in prokaryotes is because they do transcription and translation at the same time. So if you get attenuation, you can stop transcription and translation together because they're still occurring, right? Because, fuck, dude. I can't see. Because if you stop translation, transcription is still happening, so you still have time to stop transcription. Does that make sense? Okay. Like, go back.
Oh.
Yeah. So I just want to break. Can you move the little pictures on the side? Everyone's video. So just to go over attenuation really quickly, this, I think I was on Office Hours with you. I'm blanking on your name. Was it you? No? Okay, there was somebody who I was on Office Hours with, and I was like working out attenuation with them because gets so complicated so quickly, but once you get it, pretty straightforward. Regions 1 and 2 in that 1, 2, 3, 4 are complementary, right? 1 includes the trip codons, the trip codon. You can see it up there in the blue. That trip codon is where the pausing occurs, right? Because if you don't have trip tRNA, the ribosome is just sitting there waiting for trip tRNA to come in and bind complementarily anticodon to codon. If you don't have trip tRNA, ribosome's just going to sit there and wait for one to show up, right? That's how that pausing occurs. And then 1 and 2 are complementary and combine to each other. 2 and 3 are complementary and combine to each other. 3 and 4 are complementary and combine to each other. If you get 3 and 4, you stop the whole thing. The way you stop yourself from getting 3 and 4 is by taking up 3, right? You want to sequester 3 away from 4, and the way you do that is binding with 2, right? So 2 and 3 is good. If 1 and 2 bind, then 2 can't bind with 3, so 3 can bind with 4. If 3 can bind with 4, it will bind with 4, and stop the whole thing. Make sense? All right, so this is the first situation. If you have high levels of trip, regardless of tRNA, right, like we just talked about, the amount of trip tRNA is irrelevant because you have high levels of trip. So trip will be bound to the repressor, activating it in its active state. The repressor will bind to the operator, and you don't get initiation of transcription. RNA polymerase will not be able to go down. Sounds good? Okay. In the second situation, there's low levels of trip, so it's not bound to the repressor. The repressor is not active, and it's in active state. The repressor does not bind. This means that RNA polymerase can then bind to the promoter and start transcription, right? If you have low levels of trip, like we were just talking about, you might still have high levels of trip tRNA, but in most situations, you have low levels of trip because you use that to make the tRNA. The tRNA is being depleted. You probably won't have enough. So this is a situation where you have low trip and low trip tRNA.
When you say trip tRNA, you mean like a charged tRNA with like a
Yes. Yeah, I don't mean just the tRNA itself because that is like tRNA that is associated with tryptophan, but I mean like tryptophan that's or tRNA that's carrying tryptophan. The reason for that is if you have an uncharged trip tRNA, it won't like bind to the codon in the ribosome. So ribosome is still bind. So you're still waiting for a charged tRNA to show up. So in this situation, you have you transcribe 1 and 2, and then you get the pause, right? Once you transcribe 1 and 2, they kind of line together. right? The ribosome gets stuck on the trip codon, right? In that time now, 2 and 3 have bound because the ribosome got stuck. Since 2 and 3 got bound, 3 and 4 can't bind. So you get the RNA polymerase keeps going and the ribosome hops along. Does that make sense? Also do want to check the chat because I said type in the chat.
There's no chat.
Okay. Okay, perfect. Don't type in the chat anymore. Just say it out loud. How do I go back? What the fuck? Okay. All right. In the third state, which is what we were just talking about, you have low levels of TRIP and high levels of TRIP tRNA, right? Because it used up all this TRIP to make the charged TRIP tRNA, and you haven't yet used that charged TRIP tRNA to make your proteins. In this state, the TRIP is not bound to the repressor still, so the repressor is still inactive. It does not bind to the operator. You get RNA polymerase binding, you get transcription. And we to the translation part, there is enough TRIP tRNA present, so attenuation occurs because it like runs through that TRIP site because there's a lot of TRIP tRNA. It quickly diffuses in. My little assistant.
Oh my God, I'm privileged.
Yeah, so again, you have that like wait while the 1 and 2 bind, right? And then because it's able to quickly keep moving as the ribosome or the RNA polymerase moves, the ribosome is able to quickly run through that TRIP, so it gets hit very quickly on the stop codon. 3 and 4 are able to bind because 2 is not really bound to anything. In reality, what will also happen is you'll get 1 and 2 binding too. Or sorry, 2. Wait, yeah, that makes sense. 1 and 2, 3 and 4. Yeah, 1 and 2, and then 3 and 4 have now bound, and that results in this getting stuck on hitting the UGA, the stop signal, stop codon, and then this will also pop off the RNA polymerase. Does that make sense, everyone? So does everyone understand the steps, especially of attenuation? Yes.
Is it just high TRIP? Transcription is not initiated.
Yeah, so you don't get any mRNA at all. What happens is since tryptophan is bound to the repressor, it's active, it sits on the operator. And this is a question from 151. How does that stop transcription? I'll give you like a 75 for that, like 3/4, not like a 75, like not like a C, but like 3/4. The other thing is that like if you think of the RNA polymerase as like a train going down train tracks, if you have like a train cab just sitting on the tracks in front of the train, right? So it physically will block the RNA polymerase from traveling down. Does that make sense? It's just like a brick wall. It can't. Although brick wall is not much to a train, but yeah, what's up? Yes, and the reason for that is this right here, right? It's able to, because there is high levels of tRNA, it doesn't pause right here, right? Because right here, and this is the situation where there is no tRNA, like no charged tRNA. The reason that it pauses here is UGG. Yeah, it's UGG. UGG is a codon that codes for tryptophan, right? So the RNA polymerase is sitting here and waiting for a tRNA to come in carrying tryptophan and for that anticodon to bind to this codon, right? If there's no tRNA, that won't happen. So the RNA polymerase is just sitting here and waiting, right? Yeah, yeah. And the reason that is a good thing is that physically the way that it stops such that the ribosome is basically covering up one. So one can't interact with two. So two, then the only option for it to interact with is three, right? And if two interacts with three, then now three can't interact with four, which means that.
Can you clarify the difference between tryptophan and 2RNA?
Can I
let's just
erase
ch-
one of these screenshots?
Huh? Yeah, sure. Which one? This is this one. Okay. Is it this one?
I don't know. it should
I
be
swear,
that one.
I swear I heard. Close window?
Okay. Oh, wait, wait, wait. All right, I was gonna draw like chalkboard, but while Robert's figuring that out, basically tryptophan is just the amino acid, right? When you think about how translation occurs, you have the mRNA, which is bound to the ribosome, and the ribosome chug along, and tRNA is coming in, and each tRNA is bound to an amino acid, right? And that's a charged, right? If it's bound to an amino acid, it's considered charged. If it's not bound to an amino acid, it's waiting to grab one. That's considered uncharged. So that charged tRNA, so tRNA that has the anticodon that codes for tryptophan, that is carrying a tryptophan, is the tryptophan tRNA that I'm talking about here, right? Like if it was uncharged, then it's not relevant because it won't be able to bind to the UGG anyway, so the ribosome will still pause. But if it is charged, then it'll be able to quickly bind, and the ribosome will run through. Right? You see what I'm saying? So tryptophan, when I said tryptophan, it's just the amino acid. If you guys remember, tryptophan's like the fun, funky, fresh one. That's mostly non-polar, but also has an OH group like right on the end of it. So it's like polar, but also non-polar. That is just the amino acid. That's just the trip. When it's bound to the tRNA, it's
I don't know how to make it go up.
everyone's got on the trip code or trip alpha.
I don't know how to get it go up. What?
I'm still struggling with like I was trying to type it in the chat, but I wasn't gonna get it fast enough. The so the trip levels versus the trip tRNA, how those I'm still struggling with how those levels I'm shit.
Me too, dude. I don't know who's talking about. Me too.
Are you confused about
Like if there's high levels of tryptophan, how can there be low and high levels of trip tRNA? Because I think in one of your sheets you said that it could be both. And I guess-
because trip is used to create trip tRNA. So if you use up all your trip to create trip tRNA, you'd have low trip, but high trip tRNA, let's say. But there could be scenarios where you don't use up all the trip to create trip tRNA. So then you would have high trip and high trip tRNA levels.
Okay.
Like one is used to create the other. You can't, like if you used up all of A to create B, right, you'd have low A levels but high B levels. If you don't use up all your A, you'd have high A and B.
Okay. So this
It's to-
pathway is used to obviously make tryptophan, obviously, but are you also trying to make trip tRNA?
There's two, okay, the sensing, the trip repressor is sensing for trip, attenuation is sensing for trip tRNA.
But the
Oh.
result of,
Okay.
or sorry, go ahead.
So that's where I'm getting confused then. The difference between like I guess attenuation and I guess the repressor is where I'm getting confused.
Like ultimately, you just need enough trip in your cells to create proteins, right? If you have enough trip tRNA, it doesn't matter how much trip you have because it's ultimately the trip tRNA that is used to create proteins.
Okay.
Honestly, I'll be real with y'all. Just memorize 3 and 4 causes termination. 2 and 3 does not cause termination. High triple levels, attenuation. Okay, that's literally, if you're really confused about this, like if you're thoroughly confused, just memorize the outcomes, write it on your cheat sheet and move on. Okay.
Can you
Okay,
say those
let's move
outcomes-
on then.
Can you see those outcomes one more time really quick? That's
Oh my
just like
God.
3.
Okay, so there's two things that control this, right? One is tRNA and the other, or trip tRNA, right? That's tRNA that is actively carrying tryptophan. And the other one is just the tryptophan itself, right? At the first stage of control, this is where just the tryptophan itself, the amino acid that is made by this gene or this set of genes, comes into play. If you have a lot of trip, right, you have repression because the cell doesn't need to make any more. If you have a lot of trip, that trip is binding to the repressor, activating the repressor, and repressing the operon, right? Because if you have trip, you don't need to make any more. If you don't have trip, it's not binding to the repressor. The repressor is inactive, and so it's not binding to the operator. You're not repressing your genes. You're activating your transcriptional pathways, and you're going to be trying to express these genes. Does that make sense?
That makes tons of
Perfect.
sense. Yes, perfect.
Perfect. The second part is the attenuation part. That's the trip tRNA part. If you have trip tRNA, 2 will bind with 3. Forget everything else. 2 will bind with 3, and you're good. Right? If you don't have trip tRNA, 2 will bind with 3, and you're good. If you have trip tRNA, 1 will bind with 2, 3 will bind with 4, and it'll stop everything.
Okay, so that I never got before right now.
There you go.
I thought the whole thing, I thought level 1, if you have too much trip, you're gonna get attenuation. And I guess I missed this in between piece.
Yeah.
Where
That's all right. You got there now.
We're
I
here.
got there. Yeah,
Exactly. We made it.
we can move on.
Okay.
Just one quick question. Can trip tRNA bind to the trip repressor?
No,
No, trip tRNA has nothing to do with the repressor.
Got it.
it's the free trip and the repressor.
Yeah,
Thank
it's the free trip that binds to repressor. The trip tRNA has nothing to do with the translation part. It has more to do with the transcription part because that's where you. Sorry, fuck. The trip tRNA has nothing to do with the transcription part. It has more to do with the translation part because it interacts with the ribosome. Like tRNA interacts with the ribosome.
Got it. Thank you.
Perfect.
All right.
All right. Does anyone have any questions? Yes. No, you're good.
So, first, when two months, is it full mRNA? Is it full or is it like?
It's not full.
It's still, yeah, this is still going.
Like the leader sequence is completely inconsequential to the protein. Like none of these 1, 2, 3, 4 sequences are translated into an actual protein that is used by the cell.
That
2 and 3 just allows for the translation of the rest of your entire transcript. Right.
That makes sense?
Right, like back to like this picture, there's different levels. If we look, the attenuator sequence is separate from all the gene sequences.
So you're go back to a sec. your RNA polymerase will only be like about here when all of attenuation happens. If attenuation happens, then it'll pop off. If the attenuation doesn't happen, I'll keep going through all of this. And then you'll get this, the machinery to make tryptophan. It's not actually coded for tryptophan. That would make any sense. Tryptophan is something, a piece of a protein. You're coding for proteins that can then make that.
Work something positive. I don't know how.
It's non-functional. Basically what happens is that like it gets technically translated, but again, it has nothing to do with actually making trip. It's just kind of
It
there.
gets popped off, killed.
So is it just the ribosome moves down that sequence and it's perfect?
Yeah. Right, like there's redundancies in regulation because the cell isn't perfect. Like imagine, right, like, okay, with all the trip levels, sure. But there's also cases where, there's high trip levels, but the repressor doesn't bind to the operator or it dissociates for a brief moment. Like there are cases where transcription can still occur even when the repressor is supposed to be bound. Like the cell isn't perfect.
Like in the lac operon, for example, if you guys remember lac operon, when the repressor is bound, the transcription is about a 7 out of 10. Or sorry, when the repressor is bound and no cap can, right? When it shouldn't be completely repressed, transcription is about a 3 out of 10. Right? Because it is possible the repressor just happens to pop off because it has a dissociation constant. We're getting like biochemistry here, but it has like a dissociation constant, which after some amount of time, on average, it will pop off. And if that's popped off, if it just so happens that an RNA polymerase with basal transcription factors happens to pop in, you'll get transcription, right? In that like very brief window.
There's always just a little bit of transcription, even with repressors.
What is the one situation that you don't get any transcription at all? I'm gonna go for the key break. Yeah, when the repressor is on. No, that's the scenarios that I'm talking about right now. If you even if the repressor is active, let's say like freak of nature, not literally freak of nature, just molecules are bumping, it happens to bump off, RNA polymerase happens to come in right at that moment, you can still get transcription. What? Yeah, but it's just very, very, very low. It's called leaky transcription. You have it. There you go, exactly. You screw up your promoter, RNA polymerase can no longer bind at all. That means nothing is coming out of that, right? That's like a common mutation that we're with you guys. Oh.
Okay.
All right, so that's part 1 of like 4.
That was probably the most complicated. It gets much simpler. So, do you want me to pick the? I don't
You
know.
can take it. It's just, I just basically put the 2 on.
Okay. So, non-coding RNAs,
Very little of this matters.
it might come up.
Don't
Honestly,
take notes.
just keep it simple. If you really want to memorize, just memorize rRNA, tRNA.
You guys should know RNA.
I mean, yeah.
Especially after I was late and we still have been talking about tRNA for like a good half hour now. If you don't know that, I'll be very disappointed and upset. The other fun one, I guess,
CRISPR is
we
fine.
might talk about this, Exis and T6, at some point later on as to an extron 7 activation. You guys haven't already talked about that one. This is the fun one. It's CRISPR, right? Because this acts very similarly. CRISPR RNA acts very similarly to how the 2 silencing pathways that Robert's going to talk about. CRISPR RNA basically acts as a guide RNA the same way that SI and miRNA.
That's actually a good segue. So CRISPR RNA is mostly found in prokaryotes, right? That's how they defend against viruses. We have an analogous system. It's called RNA interference. And let me just do a quick read over the slide. All right, so miRNA is classically considered endogenous. What does that mean? It means that the miRNA is coded by your DNA in the cell, right? So RNA polymerase in the nucleus has to transcribe your miRNA segment to something we call pre-mRNA. And then that is processed in the nucleus by Drosha. It basically just cuts into smaller and smaller pieces that are then exported out of the nucleus. And then most of the time, what you're gonna see is it's a duplex of a hairpin structure. So in the cytosol, Dicer will cut off that end hairpin. So now you'll have a linear duplex of miRNA that now associates with RISC. RISC unwinds one of the pieces because it's double-stranded. It gets unwinded into a single-stranded miRNA. And now you have RISC bound to a what we call a guide strand. And that allows it to track down other complementary mRNA sequences. And when it tracks it down, miRNA tends to either localize it to P bodies, which are just mysterious death machines.
Yeah, it's basically just like an addict.
Yeah, or it can prevent, right? Like if you have this RISC complex binding to your mRNA, to the mRNA, it can't really get translated. Like it's blocking the ribosome from translating that mRNA segment. There's another image I have in the Kahoot that's probably a little bit better.
Yeah, there's better pictures, but there's a lot of stuff that you could draw on your sheet to be honest.
100% get something on there about the miRNA. 100% draw that
Oh,
on.
there's gonna show up, but there's a lot of stuff that you can put on your cheat sheet. We're gonna get into the signaling pathways. That's like part 4 of 4. It's only 3 parts, but we're gonna get into the signaling pathways. Some of the more complex ones. For example, JAK-STAT is quick. You guys should be able to know JAK-STAT. The survival pathway, right? There's a lot of moving parts in that, right? So that might be helpful. This might be helpful, but there's in Laura's slides, or maybe I think it's in her slides. She has that one picture of like the comparison of siRNA versus mRNA side by side. That's a good picture to have because that'll tell you where the paths like come together, where the paths are different, and what's involved at like generally each step. Beyond that, understand what parts of what are involved in what, right? Like what is doing the you're gonna get into that in a second, but like what is doing the cleaving and what is not doing the cleaving? Like when he says the RISC complex, right? What parts of that RISC is active and what's not? That's stuff that you should know. If you want to put that on your notes, you can. Right. But I think that side by side image is really good.
So again, like the reference to endogenous, it really means that, right, it's coded in your DNA. So your cell wanted to create the miRNA. This means that essentially the cell classically overall in general uses miRNA to regulate its own gene expression. And this is just more of what I said earlier.
That was the last step. Wait, Robert, can you go back to that? This is important. I remember this is on your problem set at some point. Which one of the two are like? What is the difference between mi and siRNA? One difference is that miRNA doesn't bind exactly. siRNA on the other hand does. If miRNA does bind exactly, then this is what I found. The Argonaut part of RISC activates, and that's what cuts.
So Aman is talking about how miRNA, unlike siRNA, does not usually exactly base pair, which might come up on the exam when you're trying to compare miRNA to siRNA. Okay. So I believe this is supposed to say siRNA.
Oh shit, yeah.
What is going
It
on?
says
Like,
siRNA.
okay, so it's supposed to say siRNA at the top. So we classically consider siRNA as exogenous. So what this means is generally the source of your siRNA is not from your cell. So something like a virus. And because your source is coming from outside the cell, it starts in the cytosol. So you don't have the same drosha processing as with miRNA. Instead, it follows exactly the same path as miRNA, just after the side, just after the nucleus. It gets cleaved by dicer into shorter segments without the hairpins. It binds to RISC, and then one of the strands is yeeted, and then the other one is allowed to act as a guide strand. Another difference is typically siRNA RISC complexes bind exactly to the mRNA, and this causes a subunit of RISC called Argonaut, and Argonaut has the catalytic cleavage activity. It literally will just cleave your miRNA in half, right? And if we learned anything from mRNA, right, the ends are capped. The 5', 3' caps, those protect the mRNA. If we just cut the mRNA in half, we leave it vulnerable to exonucleases that start to degrade the mRNA. So once you get that initial cleavage, that mRNA is just doomed to die.
You don't understand what Robert just said? Does anyone understand what Robert just said? Okay. No. Basically, it just cuts in the middle, right? If you imagine this as like a fully formed mRNA, what's at the 5' then? Yes, exactly. There's a 5' cap. 3' end, there's a poly A tail. That's like a time bomb. Things are actively chewing down the poly A tail, right? So the longer your poly A tail is, the longer your mRNA will stick around. But if you cut right in the middle, you can start degrading from here in both directions like that, right? Because you can't degrade from the 5' end. You can degrade from 3' end, but that's the point of the poly A tail. But if you cut in the middle, you should go that way. And then mRNA is toast.
Does that make more sense?
Can the people on Zoom hear me right now?
Yes, I can hear you.
Yeah, wait, so this, I think you already said it before, but I don't forget what you said. So for the Argonaut, what's exactly the purpose of it to cut in half? Like, why does it cut in half?
Because if it cuts in half, then that mRNA gets degraded. But it's not Argonaut that's doing the degrade. Argonaut's not gonna sit here and like take
Eat
off
it up.
each nucleotide. It just cuts it in half because that allows exonucleases to come in and just start chewing from these two ends that you've now made, right? Exonucleases can come in, start here, and just chew that way.
So it's just there to regulate, kind of?
Well, it's like what like causes cleavage, it's like the degradation of the mRNA. Right? By leaving it in that one spot, it allows exonucleases to come in and actually degrade. So that's if the siRNA binds like with perfect base pair, then Argo will make that cut, and that cut means that mRNA gets degraded. Big complex. Risk is in itself like the risk complex is like a bunch of stuff. And that risk complex, a big part of that is Argonaut.
Cell signal.
Real quick, does anyone have any questions about RNA interference?
So I thought that it was like microRNA that base pairs exactly with the target mRNA and degrade with like siRNA that like get perfect base pairing.
I think you just swapped it.
What is it?
I think you just swapped it. If you go back to the lecture videos, I think you just swapped which one was which.
Yeah, I just look it up to make sure. siRNA is usually fully complementary. mRNA is usually partially complementary. The reason that the mRNA pathway is a little more complicated because it's endogenous, right? So it's not just used for cleavage. Because if you think about why is this happening? Right?
Come on, get closer if you're gonna explain this.
Like why is this pathway happening like this, right? You have, if you imagine that this is inside of a cell, this is an exogenous piece of RNA, right? So let's assume that this is a virus in the tech cell and stuck its RNA in. And now you have this piece of RNA floating around. What's going to happen is basically happening is Dicer is seeing that going, oh shit, that's bad, right? And using that viral RNA as a template to break down other viral RNA, right? So it needs to make sure that it's actually targeting viral RNA. Because if I have an important gene that's kind of similar to a viral gene, right? And now that virus comes in and is picked up by this pathway, then my important gene is going to get knocked out. Does that make sense?
It comes down to evolutionary function, right?
Sorry, well, I just want to clarify, you look confused. I was saying that in the like hypothetical where siRNA doesn't need complete binding, right? If it's okay, if it's like a little wishy-washy, then my important gene would get knocked out. We don't want that. We want it to be specific, so it has to be 100% base pairing. Yes, now Robert.
It really comes down to evolutionary function, right? Like, why does siRNA have to be a duplex RNA? Why? Because viruses, some of them at least, are double-stranded RNA, and none of our genes use double-stranded RNA. So we know if you detect double-stranded RNA, it either has to be mRNA or it has to be a viral RNA. And so the siRNA pathway is very specific, like Aman said, because you do not want this pathway targeting random genes in your cell. This is why, like, the miRNA, that's why you don't need exact base pair matching. Like, a single mRNA potentially could bind to different types of mRNA, right? So you could have one miRNA regulate an entire class of proteins based on evolutionary function. Like, it could be like a domain in terms of their transcripts, or it could just be something else that's similar within those transcripts.
That's the kicker right there. Is that you want to win this part right here, repression, right? It's not necessarily cleaving, so it's not necessarily like bad mRNA, right, or like viral mRNA, but a lot of times it's like the cell's own mRNA that's like, actually I made that on accident, basically, right? There's too much of that mRNA around. I don't want that much of the protein. It'll use this pathway to basically just sequester it, right? Pull it out of circulation. And if it's out of circulation, it can't be made into a protein. And that can be used to shut down like a whole class of proteins.
Yeah.
So for mRNA, it has P-bodies, and then for siRNA, that version of P-bodies is the Argonaut.
No.
Close.
So the question is, miRNA has p-bodies, siRNA
Just as Argo.
has Argo, but like, the way you need to think about it is this, right? In your cell, right? Your cell does not want random strands of DNA and RNA around, so it keeps a lot of nucleases within the cytosol. These nucleases will attack unprotected RNA strands, right? So when the siRNA cleaves in the middle, right, you're leaving this gap, this exposed mRNA that allows other enzymes in your cell to degrade it. So the p-bodies are different. We won't really explain how they work because Laura doesn't, nobody really knows how they work. Let's be honest.
They can still get degraded with the same the Argonaut process, but that's
takes longer.
no, that's only if there is 100% like pairing, right? But a lot of mRNA does, or mRNA doesn't need the 100% pairing, right? So think of like mRNA, like the end result of mRNA is like an siRNA, but with like added bonus features. And that bonus features is that if it doesn't pair perfectly, it gets either sequestered and repressed just because it's like bound to something. So a ribosome can't bind to it, or it gets popped into a p-body, which the reason I said the p-body is like the attic of the cell is just that like it just stuffs it up there, and then it's just like kind of there, you know, it's not able to do it. It's not like in use, nothing is happening with it. It's just kind of there. Beyond that, honestly, we don't know all that much about p-bodies. It is possible that a p-body might be able to fuse with a like a lysozyme or some other like
Catalytic organelle.
that's used to break down a whole bunch of stuff, right? Lysozymes are just basically bags of enzymes. So if you fuse that with a p-body, you're just gonna destroy everything that's in that p-body. So you don't need to know all that much about what happens once it's in a P-body. It's just once it's in the P-body, it's either going to be just sat there or it's going to be broken down.
So mRNA is just a better version of siRNA.
It's not better, it's just different. They have different functions.
mRNA covers like more in general.
I guess
I
it's
think a fair way to think of it.
it's more flexible because the cell wants that flexibility. It's regulating itself. So the more flexibility, the better regulation you can have. The siRNA, you don't want flexibility. You want it to be strict, exact, and yeah. No problem. Anything else?
So I just have a quick question. In that diagram, all of the short spots that popped out.
She's talking about this.
Where it shows them in the nuclear, it clearpins and then this situation kind of doesn't have any.
Come on, you got an explanation?
No idea.
Honestly, no
I have
idea.
no clue. I don't know if that's specific to this diagram. That's
Well, so the other ones had it, that's what I was wondering.
I-
And then
this is real quick. It's I don't. This is definitely not something you can know, but my guess would be that it's used to, like, if you see that, like this, most likely this strand with the bump, for example, is the one that it doesn't base pair completely perfectly, right? At that location, and that's the one that's used for, that's like what allows risk to target one of those strands over the other for a combination into the risk complex.
the only other thing that I was wondering, in the diagram that I did, it showed mRNA forming short proteins and siRNA forming double strands. Is that, like, do they only do that, or could either of them do that?
siRNA can also do short. It depends on, because siRNA, like, there are viral genomes that are short hairpins, there are viral genomes that'll be double strand. It really depends on the exogenous stuff. There's like a lot more variability in what comes out of the random environment and like whatever the virus has rather than the cell itself. Yep.
We're good to move on.
Anyone else have any questions about RNA stuff?
No? Cell signaling. Signaling. I don't know. It's just how cells communicate to each other. It's not that deep, bro. Right? Like, generally, they're small molecules that we pass around to other cells and they bind to receptors. So some signaling molecules are small, nonpolar, and they can diffuse through your lipid membranes, right? But a lot of them cross,
No, I'd say diffuse.
diffuse.
I don't mean to like blow you.
Ugh. But some molecules can't, right? Like insulin. Insulin cannot just randomly cross cell barriers, cell membranes, because it's a short peptide. And so, you know, I don't know, what's on the next slide? Classically, what we see in cell signaling is you have a signaling molecule, it binds to a receptor. That receptor acts as a bridge between the outside world and the inside world of the cell. The receptor activates some protein, and that protein leads to a cascade that ultimately forms your response. And so one element of signaling is signal amplification. So, right, we can start with a couple signaling molecules, maybe 1, 2, 3. They bind to a few receptors. How can 3 receptors result in a whole change in your cell? And the answer is amplification. At each step in your pathway, that receptor might activate 100 signaling molecules. In this case, let's say it's cyclic AMP, and those 100 cyclic AMPs are able to activate each 100. So that's 100 times 100, that's 1,000. And then that 1,000 can activate maybe 1,000. Oh, it's 10,000? My bad. 10,000, maybe those 10,000 each activate 100 by themselves. And now we have some number. I can't do the math. All right, but like that's the essence of signal amplification.
Talk about how you feel about that.
Uh-huh.
I was saying that you're better at math than I am, so like I need to like really
It's okay, Aman.
grind it in.
Well, I chose bio because I don't like math. And so let's go over the first receptor, the RTK, or the receptor tyrosine kinases. Essentially, they have a receptor domain, and normally they're in an undimerized conformation. When the signaling molecule binds to that receptor, the receptor is dimerized. The receptor innately contains a kinase domain. And when you bring the two receptors close enough to each other in the dimer state, those kinase domains interact with each other and they create cross phosphorylation, right? And so now your RTK just phosphorylated itself. That phosphorylation can attract whatever proteins are in your pathway. And this is like the kickoff to a response.
Is cross correlation just the same thing as autophosphorylation? No.
So if you go back, if cross correlation means that this RTK, right, is the tyrosine kinase domain here is phosphorylation. These are, I'm gonna do that over here so they can see maybe. This tyrosine kinase is phosphorylating the tyrosines on this receptor, right? So this receptor 1 is phosphorylating receptor 2. Autophosphorylation would be receptor 1 is phosphorylating receptor 1. Right? So it's like very slightly different, but it is different.
They will, what? Sorry.
So for the molecule, for RTK, does it always have to be a dimer? Because I know because for JAK-STAT, it's not a dimer. I mean, the signal's not a dimer, but it's still, the receptor's still dimer, even though it requires one signal.
I mean, it has to bind to both receptor sites. So
But then
I
I'll
think-
go for JAK-STAT where
I
even
think
though
for
it's like,
RTKs, it depends.
both are two separate,
It's just like,
but for the dimer, they'll have one signal.
honestly, that detail, let's just pretend it doesn't really matter. Don't mention it on your test. Just don't talk about it. Just say the receptor dimerizes. Don't talk about the signaling molecule.
I think it really just depends. As far as I know, it depends on the pathway. Because I think, like for insulin, for example, I think it is individual insulins, right? They
It's
don't do-
probably a case by case scenario.
Yeah, it's
All I
a.
know is that-
So,
all I didn't know is that it's just.
yeah, there might be like two. Like it might be that this a signaling molecule binds here, and now this one's looking for
Oh,
a second activated one, right? And then another signaling molecule binds here, and now they can like do whatever. Or in this scenario where it's like one thing that binds to both at the same time.
So the RAS signaling. So it's a build off the RTK, right? So what actually binds to the RTK is going to be an adapter protein. In our case, we're just going to call it GBR2. How does GBR2 bind to the RTK? It utilizes an SH2 domain, and the SH2 domain, all it does, it binds to phosphotyrosines. And then now you have a GBR2 bound to your RTK, but it also has to interact with the next protein in your pathway, right? So it also contains an SH3 region, which just means it interacts with polyproline regions, whatever that means. And so that allows it to interact with the RAS activating protein. On the slides, it says SOS, but you can just call it RAS-GEF. Right. SOS is just a GEF. And like in the RAND pathway, the GEF allows the RAS to dissociate the GDP and allows its GTP to diffuse in. And this is what activates RAS. So the MAPK.
Wait, go back to that. Does anyone have any questions on that? Everyone understand what has the SH3, what has the PSSP, and what has the SH2?
Right. RAS has to have a PXXP region. That is what the SH3 on GBR2 is binding to.
Oh, do we have an exam?
I forgot to grab that.
Okay, that's fine.
I can bring it.
Okay, thank you.
was gonna grab them during the Kahoot, but then we moved to Kahoot. Okay. So this is the map case signaling. Essentially,
Yeah, I was just gonna ask you a little bit.
okay.
Phosphate group? Phosphorylate
Oh,
tyrosine. Oh, fuck.
closer.
Phosphatyrosine. Can you guys see this? The people in the camera people?
They can't.
Let me see.
It's very blurry.
This, can you see my stick? I'm stick.
Can you interact with the slides at all? No.
Probably, but I want
Okay.
phosphorylated tyrosine, right? Has to be tyrosine. This showed up on your problem set. SH2 binds to phosphotyrosines, not phospho anything else. Okay, so GBR2 has an SH2 domain, right? This side, this little thing right here, is an SH2 domain, so it recognizes and binds to phosphotyrosine, right? Anything that binds to phosphotyrosine, generally it's safe to say that it is SH2. There's one specific case where it's not, and I will let you know when that is. We haven't gotten there yet. So this has an SH2, right? On the other side of it here, this adaptive protein, GBR2, which we're talking about the dark blue protein, has an SH3 domain. That SH3 domain, instead of recognizing and binding to phosphotyrosines, recognizes and binds to PXXP, which is, what is it, polyproline,
Polyproline region.
which is a region with a lot of proline amino acids, right? PXXP polyproline is on the SOS or the RASCAF. That makes sense? Yes.
Is that the GBR?
Yes, the dark blue is GBR2.
And then on the left, that's the PX.
So on the right, do
I
you
mean,
mean?
the right.
Yeah.
So yeah, this has a PXXP. This has an SH3 which binds to PXXP and an SH2 which binds to phosphatase.
So it has two. It's really the only one that will have two, I believe. Are we good? So the map K, we begin with the RAS. So the activated RAS from earlier, and this is key. Do not say RAS phosphorylates anything. RAS is not a kinase. It just activates. How? You don't need to explain, but you just need to say active RAS can activate something. And in this case, we're going to call it map KKK. Map KKK itself. Oh, map 3K itself, it is a kinase. So map 3K activates map 2K. Map 2K is also a kinase that activates map K. Map K itself is also a kinase. It's very degenerate, but right. Do you understand why there's 3 different map Ks, right?
Yes. So this one will activate this one, which will activate this one.
You have so many components in this pathway that just activate each other for seemingly no reason, because it allows for amplification, right? Within each step, each map K can activate more map Ks on the next step, which leads to a bigger signal at the very end.
So this can activate like 5 of these, which will activate like 5 of these, which will activate 5 of these. You get 5 times 5.
And so, yeah, at the very end, your map K, the final kinase, it finally phosphorylates / activates some protein, some transcription factor, and those transcription factors and proteins lead to your ultimate response, right? You like the question here, what are the possible final results?
That's a question for you guys.
Let's get an answer. What are possible final results? All right, so someone said change in protein activity and change in gene expression.
I don't see anyone get that wrong. That was on the problem set. That was on this. That was also on something else. I feel like probably. But that is super important. What is the end result of signal any signaling pathway? What is the point of signaling? You either change protein activity or change gene expression. And this part right here, we have an example of gene expression that we're going to get to in a second. That's JAK-STAT, right? That's a STAT is a transcription factor. changes in protein activity. This one right here is why amplification and multiple steps is so important, right? Because we're talking about a cell and you're trying to change protein A, and there are 400 protein A's running around in a cell. You need a whole crap load of whatever this is to change every single one of those 400 protein A's, right? Just to get like diffusion, random diffusion to increase your chances, you're probably gonna want like at least 1,000 of these for 400 of these, right? Hit every single one of those 400. If you have 1,000 of these, then you need like at least like, if it's, what's a good 1 to 3, right? Or 1 to 10. You want like 100 of these. If you want like at least 10 of these, right? So this will have to activate 10 of these. This will have to activate 100 of these, 1,000 of these. Hit all 4. Does that make sense? Yes.
I guess first one. So RAS doesn't phosphorylate anything. It just activates the map.
Yes, RAS is not
How
a
does that work? Robert said something about this. How does that work?
Would be that G. Nope, it just activates.
I don't know. Nobody knows. We haven't figured out that. Right now. That's
Yeah,
like a
so
work in
do
progress.
not say RAS is a kinase and phosphorylase, just say it activates.
Phosphorylate each other.
Yes, because that they do, right? That's what kinase means, right? Kinase means that they'll phosphorylate. So the MAP kinase means that it is the kinase that does its kinase activity on MAP kinase. Right? So this MAP kinase just means that it is the kinase for the MAP, or MAP, whatever MAP is, right? Whatever MAP does down the road. That make sense?
Yeah.
So MAP kinase will phosphorylate in 2 places. This MAP kinase will phosphorylate in 2 places. MAP kinase will phosphorylate something else. Right? Keep in mind, this is not one strict thing, right? What we're talking about here, the MAPK cascade is very loose and very general, right? This is like kind of what happens. When we're talking about specific signaling pathways, then you get into specific proteins here, and stuff like that. A lot of crosstalk happens and stuff like that, right? So it gets complicated really quickly, but this is what we're talking about like a general case. You can have a million of these, right? MAP kinase, whatever. Right? It really just depends on the specific pathway in question.
Are we gonna be like expecting to know what pathways like what
No.
I don't think so.
besides the survival pathway?
Survival pathway, the only other one I would say is JAK-STAT.
For what?
JAK-STAT,
Immunization?
STAT is a transcription factor for genes solely related to lactation. Right, so it's repressive.
It does more than just lactation. It's just one example.
I think that's like its main function.
It does stuff in the immune system too.
You said it ended in like no protein in the video.
That's like the big thing.
It's like immune cell signaling.
Yeah, I think it's true, but I think stats, there's multiple stats. I think we're talking about JAK1, then it's stat 5,
Okay,
which is stat 3 is.
we'll just live with it.
You know what I'm talking about? There's different stats. There's stat 5, stat 3, and all the different things. So yeah, it gets complicated really quickly. So we're talking about general cases here. The general for stat just goes much faster. All right.
Oh my god, I keep forgetting.
Anyone have any questions about that? Anyone else? No? Okay.
All right, we're on to the big stuff.
This, I think you should, if you're gonna put something, draw some signaling thing on your thing, I would just go with this. There's a lot going on.
So we begin our journey back at the RTK. When you receive the survival signal, the RTK cross phosphorylates, and instead of GBR2 binding to the receptor, it's PI3K. This is for a PI3 kinase. It's not.
It's just. Oh wait, shit. Never mind. Never
really
mind. I
says
thought
it
this was
there.
JAK-STAT. Never mind. Never mind.
All right,
I'm
perfect.
so sorry.
So what you need to understand between PIP2, PIP2 and PIP3, those are phospholipids. They're specialized phospholipids. That's why when you look at them, they have this little red portion that attaches them into the membrane. PIP2, this is how you differentiate. P2, it stands for 2 phosphates. P3 stands for 3 phosphates. So when you're in this 2 phosphate form, it's inactive. This PIP3 state, the 3 phosphate, that's its active form. So when PI3K is able to bind to the RTK, it becomes activated. How does PI3K bind to the RTK? What domain? Speak louder. I'm getting SH2, exactly. PI3K is binding through an SH2. When it phosphorylates PIP2 into PIP3, PIP3 is now able to become activated and it binds to PK1 and AKT. How does it bind? It binds through the pH domain.
So that's the scenario where you're binding to a phosphated something that we talk about. Where you're binding to a phosphated something that's not an SH2 domain. It's a pH domain, right? pH not isn't like concentration of H plus, but like it's just what it's called.
Okay. And so AKT, realistically, it requires dual activation, first from PK1 and then from PK2, which in this case is called M4. Just ignore that. We can just call it PK2. But like, right, there's a sequence of steps. First, PK1 needs to be bound to PIP3. Now PK1 is activated. Once PK1 is active, it can phosphorylate AKT. AKT also needs to be bound to PIP3 to be activated. But once PK1 activates AKT, PK2 comes in and gives the second phosphorylation step that allows for a fully activated AKT. AKT itself is also a kinase. What it does is when it's active, it phosphorylates BAD. BAD is a protein in your cell. Its role is sequestration of BCL2. I know, big words. So let's just pull back, big overview. Like, what is the purpose of survival signals, right? Yeah.
Does that AKT population of that, is it like that's what normally won't happen? So the standard is apoptosis.
Yeah, the standard is apoptosis, right? So like when we go back, right, like the cell, the signaling cell, what am I saying? Cells only want to be alive if there is a survival signal, right? So their default is death. How does this happen? Well, there's a protein called BCL2. It's localized on the mitochondrial membrane. When that BCL2 is active, it's able to prevent apoptosis. But when BAD sequestrates BCL2, apoptosis will occur. The only way to stop BAD from binding onto BCL2 is phosphorylating BAD, right? So in constant activation by survival signals, AKT is continuously active and it continuously phosphorylates BAD. So BAD continuously cannot bind to BCL2 and your cell survives, right? But the second you stop receiving these survival signals, AKT becomes inactive, BAD reverts to its unphosphorylated form, it sequestrates BCL2 and your cell dies in the absence of survival signals. Questions? Yes.
So if there's a loss of function of AKT, will the AKT still like dissociate from the PIP3?
All right. So the question was, if PK2 is inactive, will AKT still become active and dissociate from PIP3? No. It requires dual activation to get to that phase.
So it will just stay there.
It'll
The
just
path will just stop.
Yeah, and it also requires it in that order. It requires it from PK1 followed by mTOR, right? If you want to get into mechanics of that, basically PK1 phosphorylating opens up an active site for mTOR to go.
It's a sequential model.
Yeah.
Yes.
So if you increase the percentage of bad BCL2, would it still inhibit apoptosis or no?
So the question is, if we decrease the affinity of bad for BCL2, would apoptosis still occur? So your question is really coming down to what is even the function of bad, right? Like when bad is bound to BCL2, your cell will die.
Wait, what?
I don't,
Yeah, no, that's when that is not bound to BCL2. It binds something called the 1433 protein. That's just like a placeholder for BCL2 and it's phosphorylated. When that is phosphorylated, you don't have to worry about that. The
Oh.
important part is this part.
sorry, I got mixed up. Right, if bad loses affinity for BCL2, even when it's unphosphorylated, bad will not bind to BCL2. So your cell will survive regardless of survival signals. Does that make sense? The question was if bad loses affinity for BCL2.
Oh, yeah.
Any other questions? Yeah.
Where does P10? I remember seeing.
Okay.
Slide, but
So P10 is not on this slide, but it exists in the cell. It's a phosphatase, right? So why does P10 exist? You want to, once you phosphorylate PIP2 into PIP3, right? You need a way to convert PIP3 back into PIP2. Because if you never do this, even if the survival signals stop, PIP3 will continuously activate AKT and PK2, and your cell will not die. So your cell needs to periodically dephosphorylate PIP3 into PIP2. Yes.
so it's similar to. So it's like another.
Yeah, it's the functionality is the same. It's just a different system, right? This case is phosphorylation, dephosphorylation. And like in all cascades, right, there are phosphatases active constantly because you want to stop the cascade once the signal stops. Any other questions? Yes?
Yeah.
Yeah. Jack, stat.
Okay, so. Is this what I'm talking about? I don't remember.
Let me get out the way.
But, okay, so this is a Jack stat pathway. They can see the slide.
I know.
Yeah, so you have these receptors. They're labeled as prolactin receptors. I don't know off the top of my head if you actually need to know what they're called. But the most important part of them being prolactin receptors is that they're not RTKs. If you say that they're RTKs, that's very bad. You'll get points off, right? Because they are just receptors, right? They don't have any kind of kinase domain. An RTK is a receptor that also has a kinase domain for tyrosine. If you remember what a MAP kinase is, the kinase, something that phosphorylates MAP. So a tyrosine kinase is something that kinases phosphorylates tyrosine. So a receptor tyrosine kinase is a receptor that can phosphorylate tyrosine. Here, the receptors can't phosphorylate anything, right? Jack can phosphorylate things. So Jack is a kinase, but it is distinct from the receptor. Does that make sense?
As an accessory.
Yeah. So JAK1 and JAK2 are each bound to their own receptor. Similarly to RTKs, but not RTKs. Once the signal has bound, the receptors will dimerize. Once the dimerization occurs, right? Once that dimerization occurs, these cross-phosphorylate each other. They cross-phosphorylate each other. So this JAK will phosphorylate this JAK will phosphorylate this JAK. It bothers me that these are labeled JAK1 and JAK2 because JAK1 and JAK2 are actually different jacks. There's a whole family of jacks. But this is the first jack, this is the second jack, and they phosphorylate each other, right? Once they phosphorylate each other, they're both active. That's what this little bump means. They're both in the active state. So prior to that phosphorylation, they're in the inactive state. Once they're active, they each phosphorylate their own receptor, right? So it's not cross phosphorylation the same way that the RTK does it, because the RTKs each phosphorylate each other. In this, the jack is phosphorylating its own receptor. That make sense? Okay. Once it phosphorylates its own receptor, that acts as a binding site for the SH2 domain of stat, right? But these are stat monomers. So once each stat binds to the receptor with its SH2, the phosphorylated tyrosine on the receptor, they are phosphorylated themselves by the jack, the active jacks. Once they have been phosphorylated, they can come off of that phosphorylated tyrosine on the receptor. And these phosphorylated monomers then dimerize to make an active stat dimer. This is a stat 5, right? You don't need to know the 5, just know it's stat. We don't talk about the other stats in this class. Once stat dimerizes, it essentially works as a transcription factor that enters the nucleus with the help of importin, which we talked about last exam. And yeah, it acts as a transcription factor for lactating related proteins.
There's a question on that.
I have no idea what you just said. Is that it?
Okay, so there's a problem like that. It's a problem with that. I'm sure about. I forgot the like idea.
Okay.
And it was like, what would not happen if Jack not be phosphorylated?
Right.
And then it's like, if Jack not be phosphorylated, then they would still be able to dimerize somewhat.
Yep.
But would not remain dimerized because it's unstable state.
That's fair.
And because they cannot stay in a dimerized state, in stat 5 cannot be phosphorylated.
I wouldn't say that it doesn't dimerize because the signaling molecule is still bound, right? What controls the dimerization is the signaling molecule binding. The JAK phosphorylating, right, means that if the JAKs can't phosphorylate, if that ability is cut out, there's a loss of function of the JAK, what it means is they can't cross phosphorylate, which means that they'll stay inactive. So the JAKs themselves never get phosphorylated, they stay inactive, and then because they are inactive, they can't do this, they can't phosphorylate the receptors, they can't phosphorylate the stat. Stat won't bind at all because receptors aren't phosphorylated. So you won't get stat binding, you won't get stat phosphorylation, you won't get stat dimerization, and you won't get the transcription factor.
Okay, so it has nothing to do with the stability of the receptor.
No, because that's based on the prolactin. Because JAK does not have to be phosphorylated to cause the dimerization. Right? Did everyone at home understand that? There was a question on the problem set about what happens if you have a loss of function in JAK's phosphorylating ability. Basically, from this point on, or this point on, everything gets screwed up. It'll still dimerize, but none of the phosphorylating stuff will happen. Yes. Yes, like the specific proteins in milk, right? It transcribes specific proteins in milk, but I would hazard saying milk because milk has a lot of stuff in it. But specific milk proteins. Yeah. Yes.
Since that phosphorylates the receptor, is that polyphosphorylated?
No, because of just the JAK is distinct from the receptor, right? The same reason that it's not considered an RTK because they are separate things. JAK is not autophosphorylated. So 2 questions, the orange ones. What domains allow the stats to dimerize? You can answer this at home too if you'd like in the chat. Robert's monitoring the chat. This question. What allows the stats to dimerize once they've been phosphorylated? What? Nope. Who said that? Is that you? Don't wish for that. Loud and proud. Let's hear it. There you go. Wow, I sound like such a dick right there. I apologize. But yeah, it's SH2 because what's actually being phosphorylated on these are tyrosines, right? The stats are having their tyrosines phosphorylated. So when this is binding to this, it's binding to a phosphorylated tyrosine on both at both sides. So the thing that binds to phosphotyrosines, as we know, is SH2.
Another question you could ask is like, how is the stat dimer entering the nucleus? What happens to the stat dimer that isn't true about the stat monomers? Okay. We have a question in chat. If Jack lost their ability to phosphorylate, would stat not be able to dimerize?
Yeah,
Right, because the dimerization occurs from the SH2 domains interacting with the phosphorylated tyrosine on each stat.
exactly. So if Jack no longer can phosphorylate, it won't phosphorylate the stat, and the stats won't recognize the phosphorylated regions on each other. Make sense? Yep. Yeah. Yeah. No, so that's these ones. That's the phosphates that are on the prolactin receptors are being recognized by these stats, right? And they state these phosphates are associated with the receptors, not the stats, right? And then these phosphates are added by the Jack. Does that make sense? Yeah. Does anyone know what so what is Jack? Because the receptor does not have a kinase domain. What is Jack? Does anyone know what Jack stands for?
I hear whispers. You gotta speak up.
There you go. Just another kinase. Literally just somebody got bored and they're like, it's a kinase. That's it. That's the entire name. It's just another kinase. So the Jack is doing the kinase activity, not the prolactin receptor. Make sense? Okay. What? Yeah, okay. No, yeah. We're not going to talk about that. It's boring. They do have another name now, by the way. I don't know who said that. They do have another name now. Just fun fact. They're called Janus kinases, but that's boring. So we're not going to talk about that. All right, last thing is GPCRs. GPCRs are G protein coupled receptors. The GPCR is just the black squiggly thing, right? Not any of the rest of it. This is the G protein. This is the G protein coupled receptor. Yes. Yes. Oh shit! Yeah, we never actually. Your question about how does it get
Yeah.
into the thing?
Well, yeah, but what is important binding on the stat 5 diamond?
Actually,
All right, like there has to be an MLS on the stat dimer that isn't revealed on the stat monomer. When it dimerizes, right, the NLS is located on the outer shell of your protein. Something about that dimer formation reveals an NLS on the outside of the dimer. And that is what I wouldn't say moves
it's
it,
probably an interface with like part one and part the other.
right? Like, but it has to be something about the dimer that the monomer lacks, which could be like an interface where it's both, or like, I don't know, it could be a lot.
Could be like something moves to the inside. Could be a
Yeah, it could be a
confirmation
movement.
shift. I don't know.
But there just has to be an NLS on the dimer.
Does anyone know? Everyone knows what NLS means. Yes. Of course. All right, back to GPCRs. This is the GPCR, this is the G protein. G protein coupled receptor, G protein. G protein is coupled to the receptor. In the inactive state, that's what you see up at the top. That's a resting state. The G alpha subunit of the G protein is bound to GDP. Once a ligand binds the GPCR, the G protein coupled receptor, right here, you have the G protein coupled receptor acting as a GEF. Right, I forgot that I want to ask you guys. What does a GEF do? Close, how does it do that? The first half was right. Second half was not something that it does. Dissociated, yeah. So, or diffuse. So the GPCR, the receptor itself, has GEF activity. So because it has GEF activity, it knocks the GDP out of the binding pocket of the G alpha. Once it knocks the GDP out of the binding pocket of the G alpha, a GTP can dissociate into that binding pocket, right? You guys had questions on the problem set about what happens if you screw up the affinity for the alpha subunit to GDP or GTP or whatever. Expect things like that on the exam. Once GTP is diffused in the G alpha's binding pocket, it's become active. That means that now the G alpha can like go off and do its thing, right? It can do a number of things. It can activate a map cascade. It really just depends on the specific signaling pathway. It can do a whole bunch. When the G alpha goes off and does its thing, the beta and gamma subunit, which still stay together, right? They don't dissociate from each other. They can also go off and do their own thing. And the beta gamma subunit will also possibly activate its own thing. Usually beta gamma is used for like membrane related stuff. So like ion channels and things like that. And alpha is used for cascades, right? Perfect. Then how does it get deactivated? Eventually something called an RGS, a regulator of G protein signaling, binds to the G alpha and activates hydrolysis, right? So it acts as a gap, which means that it doesn't actually hydrolyze the GTP itself into GDP. It activates the G alpha's innate ability to do so, right? So the RGS cannot hydrolyze GTP, but the G alpha can, and it will only do so when bound to or activated by RGS. So RGS will speed up the process by several million times. What? Yeah, lots of magnitudes. So that's RGS functions as a gap. Then once the G alpha is GTP bound again after carrying out that hydrolysis, the G alpha, the G beta, and the G gamma, and the GPCR come back together. Question? Excellent. Oh, okay.
Warren mentioned that gamma and G alpha, like they could be violent because they have.
Yeah.
Is that like influence this universe?
No, but that influences what it does, right? You know how I said G alpha, I'm pretty sure comes off. It can come off the membrane once it's active. Beta and gamma can't, right? So they still have this lipid tail. I don't know if you see this like little black line there that keeps them anchored to the membrane, and that's why they generally interact with membrane-associated things. While G alpha is considered like the main thing because it can go off and do a whole bunch of stuff, while G beta and gamma are limited to the membrane. So that's why beta and gamma usually work like ion channels and stuff like that, like bringing calcium ions, potassium ions, sodium ions in and out of the cell, or like other channels, receptors, things like that, as opposed to downstream stuff. Yeah. Does anyone else have any questions? One chat. Do we need to know how G beta and gamma is deactivated? Is it when G alpha goes back and binds again after being deactivated? Yes. Amber, that is spot on. Second half of that is the answer to the first half of that. G beta and gamma is deactivated when G alpha goes back and binds again.
You probably want to.
No problem. All right.
So the end goal is
What's the second step? Oh, you mean in the third out of these four? Oh, yeah, this goes and does some signaling cascade. But the thing is that it's not turned off by carrying out that signaling cascade. It's turned off by the OJS. Right? That makes sense? Can you go over some SH3 domain examples? Yeah, you only really have to know one, and that is this one right here. It's on the RAS activating protein. Or sorry, it's shit. Yeah, it's on GRB2, and it binds to the SH3 on GBR2, GRB2 binds to the PXXP on the RAS activating protein. That's the only SH3 domain that you guys know. Does that make sense? The slide. Nicole. Nicole. Oh, okay, perfect. Excellent. I think so. All right, we're gonna go. Robert's gonna go over some questions from the what's it called? From the Google form, and then I'm gonna give you guys some tips on how to ace this exam, and then you guys can all go home or go get dinner or whatever else you want to do. Oh shit! I mean, poop. We also have a Kahoot, and that'll be fun if you guys want to stay for that. I know we've been here for a super long time, so like feel free to like get up and stretch or do whatever, and what diagrams will be given to us? Did I answer that? What diagrams will be given?
At most, the amino acid chart and the RAND, but you might not even get those because they're not gonna come up.
Yeah, the amino acid stuff is probably not going to come up. But beyond that, there's not really much in terms of diagrams that are given to you. Is RGS not considered enzymatic? Why is RGS
Not considered enzymatic.
not considered enzymatic? I think we went over that. Did we do that? What?
Well,
RGS is not considered enzymatic because it doesn't actually hydrolyze the GTP to GDP. It makes the enzymatic portion of G alpha hydrolyze the GTP to GDP. Done pretty much most of it. Not a question, but I don't believe the Zoom link is working. That's my third one. Do RTKs fall under the classification of proteins? Yes. Does the Jack kinase subunits? Yes. Everything we're talking about right now is proteins. Apart from the proteins that are lipid anchored, and those are still proteins, they just have like a little lipid piece. What is tRNA? We discussed that. If you guys still have questions, feel free to ask, and I can like draw it out for you. Perfect. We don't have a second back here. The one last thing. Hold on. Can we go back to the slides? Okay.
Oh, sure.
Right. So one last thing. Recipe for success before we move into the Kahoot. So I would suggest, this is for studying, not necessarily for your cheat sheet. Draw out what happens to trip operon in each state, right? Like literally draw a strand, a squiggly of DNA, draw a little promoter, an operator, the gene, a binding site for, what's the other thing? Yeah, that's really it, right? Draw all of that, and draw what happens when you have these three states, these three stages. I want to move this. When you have high trip and low, high trip and anything else, high trip and low trip tRNA and low trip and high trip tRNA, right? Draw those three states out, draw what's bound to the DNA, what's not bound to the DNA, and what is the end result? What happens to RNA? What happens to attenuation state? Right? If you can go over all of those three states of trip and tRNA and understand what that means for what's bound and not bound to the DNA and or mRNA, and understand how that affects the outcome, you'll be all set on the trip operon. And then for both the signaling and non-coding interference, non-coding RNA interference pathways, go through each step in the pathway, like take like one of those diagrams. Like you can pick one of the ones from this slideshow. Just don't look at a slide with words or like delete the words or something. I don't know. Find a picture on Google and walk and talk yourself through every single one of those steps. Make sure that you know those steps, right? Really well. Know when Dicer is coming into effect, know when Drosh is coming into effect, and know what those things do and how those things work. And then the last thing, I've been asked several times how to answer the questions, and I made 5 steps for that. Right? I've only taken one class with Laura.
I've taken 2,
Yeah.
61.
Yeah, it was the only class I took with Laura.
Oh, shit.
I actually didn't take 285 in this department. I took it in the back end department. Step 1, but I know how to answer Laura's questions because I did that for a long time and I grade those questions. I grade all of your problem sets. Step 1, identify what the question is actually asking, right? The question is, this is a 151 question that I wrote this from. Where does the tRNA bind? Where in the ribosome does the tRNA bind when it's bringing an amino acid in? The answer, if you remember EP and A sites, the answer is the A site, right? But so the question is asking about literally where does the tRNA bind when it's bound, right? That's step 1. Step 2 is why is Laura asking this question, right? Laura's not asking you these questions to see if you know the P site or the A site, right? Laura's asking any of these questions so you can demonstrate your knowledge about the specific topic that she's asking about in the context and to the level that we covered it in class and like in review sessions and in homework and problem sets and whatnot, right? So you have to demonstrate your knowledge. This is why don't go by the answer key when it comes to how much should I answer for each question, right? Because the answer key will do what one does, right? The answer key will answer each question the level of step 1, not the level of step 2. Step 3 is answer the specific question, right? That you identified in step 1, answer that. The answer to the example I gave you is the A site. There you go. That's step 3 done. Then add whatever else is needed, extra context, extra information, extra things that happen necessary to demonstrate your knowledge. What I would go with is what is involved, how is it involved, and why is it involved? So for that question about what part of the ribosome does the tRNA bind to, or like what pocket, I would say the ribosome binds to mRNA, right? The codon on the mRNA will bind to the tRNA. The ribosome is bound to the mRNA such that the codon on the codon on the mRNA will bind to the anticodon on the tRNA, and that codon is sitting in the A site of the ribosome. So when the anticodon of the tRNA comes in to bind with the codon on the mRNA, it will bind in the A site. I added a whole bunch of words, and that was not super well spoken, but I showed that I understand that the mRNA is where the tRNA is binding, it's what the tRNA is binding to. I showed that I understand that it's the A site of the ribosome, so I answered step 1. I showed that I understand the way that the ribosome and the mRNA interface. I showed that I understand the way that tRNA and mRNA interface, and I understand why all this is happening together. Does that make sense? Right? Because when it comes down to what we do to grade your problem sets, when I grade your problem sets, and when we grade your exams, we're not grading from the answer key. We're grading from a rubric, right? And that rubric has like 3 things that we have to see. Most of it is like, did they mention this one thing or did they not, right? Is the question right? That's 1 point. Did they mention this other thing? Yes, that's 1 point. Did they mention this 3rd thing? Yes, that's 1 point. So if you write 8 things and you only need 3 of them, you're still gonna get those 3 points. But if you only write 1 thing you're gonna miss out on 2 points. Does that make sense? Last step is go back and make sure that nothing that you wrote in step 4 is wrong. Because if you write 8 things and we only need part 1, part 4, and part 6, but part 5 is just egregiously wrong, we're gonna have to take points off. Right? Even if it's not necessary for our answer, even if it's not in the rubric, if it's just straight up wrong, we have to take points off. Right? So make sure that nothing you write in part 4 is wrong, but make sure that you do part 4. Right? Don't let step 5 keep you from doing step 4. A good way to practice that is if you guys know people in the class, or if you guys have study buddies, answer the problem set, right? Go back and do a problem set without the answer key, without anything else. Add all the extra information, right? Add everything in step 4, and then give your answer, or like give your sheet to your study buddy, and you take your study buddy sheet and you read their answer and say, does this make sense? Is everything in here right? Right? You fact check them. And that'll tell you if you're doing step 5 right. That makes sense? Go back and redo your old problem sets. Go back and redo your old quizzes. I understand that obviously the exam is tomorrow. We don't have a ridiculous amount of time. Nobody actually wants to study this. Nobody actually wants to redo their problem set. But that being said, it is the best way to study, right? Laura makes her exam questions from the problem set. Does that make sense? Is that helpful?
Do we have a time limit?
No,
No, I
the
mean,
time limit is like,
technically 3 hours. I think on the final exam, so for 161, which is an allegory to 161, it was the class that I took with Laura, class that Robert and I first TA'd together. The only person who came anywhere close to that took the exam twice. Twice, right? Twice in 2 different colors of pen. Right? She went the entire thing with black pen and then went back again to the entire thing with blue pen. It's freaking psychotic, but also she aced the shit out of that exam. But that was the final exam. She like perfect score, like perfect, amazing, whatever. But she had enough time to do that twice, and she like barely scratched the 3-hour mark. Right? You will have enough time to go through all of this for every single question. I guarantee that. And if you don't, we're told when we proctor, right? If somebody's pushing up against the 3-hour mark, just like let them know. We're not going to take away your test, right? We're not going to dock points, but that 3-hour mark is more so that we get to go home or something.
we'll be flexible. As flexible.
Robert will leave. Robert will take.
Oh, I will not leave. I don't leave. I just sit there.
Robert will watch you. for
I'll
the
stare at you.
rest of your life. Robert will sit there and just whisper the word. What is it? Diffuse.
Defuse. My number 1 tip for the exam is make sure each of your answers are self-contained, because we're grading off a rubric, right?
yeah.
There are certain points that need to be hit. You might have hit that in part A and then you just completely dropped it in part B. If I'm grading part B and I just completely forgot you put it in part A, I'm not going to consider that part of part B. Do you understand? Like, you want to make it as easy for your grader as possible. Make sure every single answer, when you read it, makes sense by itself. Make sure your questions don't depend on the last answer you just gave, if that makes sense. Especially if it crosses questions and it's not even contained to a subpart, because we're grading per question. Like if you wrote something question 3, I'm grading question 4. I don't know what you wrote for question 3. I'm not going back. Sometimes like I have a part A, part B. I'll read your part A and I go to part B, and then it's just missing from part B. I'm like, oh, I think they said it in part A, or maybe they didn't. Like, you don't want to make me check, essentially, because I might not check. I might just be like, I don't know what he wrote in part A. There we go.
All right. Does anyone have any questions about that or anything that any of us has read?
Oh, oh.
Oh yeah, I thought this was funny. I thought it was good. There we go.
All right.
All right, everyone playing at home, everyone playing in here, we have a Kahoot. We will answer questions that you have after this, like based on this Kahoot or anything else from the content or anything else after we get through this Kahoot. If you win this Kahoot, you will win extra credit in the eyes of me and Robert and nobody else.
Huh? 22, 22. Are people still trying to join? Alright. We're just gonna start soon. Yeah.
In 10 seconds, we're gonna, oh. In 10 seconds, we're gonna start. Who wants to count down?
We're done. You got it. But you can't see shit.
What?
Oh, it's kind of hard to see.
What? Oh. Can't see the question, but
Oh.
so this is PRI, mRNA into pre-mRNA, and that's Drosha, not dicer. Wait, is that right? Yeah, that's Drosha, not dicer.
So drosha is in the nucleus. It processes your pre-miRNA. And that mRNA is then exported into the cytosol where it's processed by dicer next. Oh, no. I forgot. I keep forgetting to listen. This is 17. We're gonna be out here in 30 minutes. Oh no, we don't get to leave. Why would we? I haven't eaten all year. I'm on eating Robert time. I didn't eat dinner last night. All I ate today was lunch. I'm trying to get skinny. Oh, thank you.
Yay!
Hey!
Well done. Argonaut. Who's Mary? Pop off, Mary.
It's a pop-off game right now.
You can also
MPP.
stop the recording anytime.
Oh my god, perfect. We got it. You cannot ubiquitinate mRNA. It can only occur to proteins. Not yet, calm down here. Okay. I have a list.
No, just RTK inhibitor means that there's essentially a loss of function in RTK itself. And you're stopping the RTK from doing what the RTK does. Doesn't really matter at what point along that, but it's a phosphorylation and also by a sailing molecule. Wow.
Wow, we're split. Okay, so a cell with a gain of function RAS is treated with an RTK inhibitor. whenever you're posed with these dual mutation questions, identify which mutation is downstream because that is almost always dominant, right? So what is downstream in the signaling path? Does everyone know what downstream means? Right, like further along in the pathway. Is RAS downstream of the RTK or is RTK downstream of RAS? Exactly. So RAS occurs after the RTK. So even, let's say in this case, right, the RTK inhibitor prevents complete phosphorylation. So your RTK is inactive. Is your RAS active or inactive? All right, yeah, exactly. So your RAS is still active, even though the RTK is inactive because RAS is downstream. So even if you try to inhibit the RTK, RAS is still gonna do its little thing. It's gonna activate map tree K, and then map tree K is gonna activate map 2K, and then blah, blah, blah. Does that make sense? There's gonna be a lot of questions like that. So. I know. Yes, no, that's. Well played. Your math is gonna die. So again, like the same concept, we need to think about what is being inhibited. So there's a mutation with GRB2. So GRB2 cannot bind to RAS. That would be like an inhibited GRB2. Can MAPK be phosphorylated? Right. MAPK has to be phosphorylated because MAP3K is active, right? MAP3K is downstream of the GRB mutation. So it does not care, like anything after the MAPK mutation, nobody cares about the GRB2 at that point. So if MAPK is after MAP3K, it does get phosphorylated, it is activated. The cellular response is activated, yeah?
Instead of those mutant map with a thing with a and then map not
Yeah, so if we swap MAPK with MAP3K, then MAP3K would not be phosphorylated. Right, like it's whatever comes upstream of this gain of function mutation. There's a lot of questions like these, by the way. I think she sounds good. So P10 is the phosphatase that turns PIP3 into PIP2. Yeah. So it inactivates it. Yeah. Oh, we're good. That's pretty good. So, right. If AKT cannot bind to PIP3, it cannot become activated and phosphorylate back, which is the crucial step that allows the cell to live. So if that cannot occur, the cell would die. Huh? Sorry, what? So in this question, the mutant AKT cannot bind to PIP3. That's a crucial step to activate AKT, right? So if AKT can never be activated, BAD cannot be phosphorylated, which is the step that allows the cell to live. So your cell would die. Without active AKT, your cell dies. Yes.
Why is Oh,
Yes. And green. Green is true because BAD would unphosphorylated BAD binds to BCL2, which causes your cell to die. And PK1 would still be active because, right, this is where we have to break down upstream and downstream again. So upstream is the mutant RTK. So your RTK is constantly active. So it activates everything past the RTK. So the RTK, PI3K binds, PIP2 is converted into PIP3. PIP3 binds to PK1. So PK1 is active. But now we have this mutation with AKT. So just because PK1 is active does not make AKT active. Right? So this is a mutation where there's a gain of function upstream, but there's a loss of function downstream. And when this occurs, the response is not seen. Does that make sense, Amber? If you're struggling with these problems, I guarantee you, Laura is going to put a question like this up, but you're going to have infinitely more time than a minute to answer them. She's going to have a question with some hypothetical mutation. There might be one mutation, there might be two mutations. And then she's going to have subpart A, B, C, D. Would this occur? Would this occur? Would this occur? Would this occur? Yes, no, why? So you need to, yeah. Okay.
But if this can't be phosphorylated, right, then the RTK is constantly doing stuff that would phosphorylate PIP3. So you have PIP3. Remember that the two things that bind to PIP3 are PK1.
It's PI3K.
And AKT. So PDK1 is still binding to PIP3. It's AKT. Oh, yeah.
So again, Figure out which mutation is upstream of the other. The upstream mutation occurs, it affects everything downstream of it until you hit the second mutation, and that mutation affects everything downstream of that. There's like it's two parts. Okay, A is not true because the GPCR is not involved in the survival signal. The blue answer, right, G alpha is downstream of GPCR. If that has a mutation that lowers its affinity for GTP, G alpha is less likely to be active. So a weaker than normal response should be observed, which is what the yellow response is basically saying. And then the green is not true because, right, if G alpha is not even active, then you don't need a gap to deactivate G alpha.
The other thing is like, the point of the gap is to go from GTP band to GDP band, right? If you're not, if the T alpha doesn't have affinity for GTP, what happens is it's not that the GTP becomes GDP, it doesn't hydrolyze, it just comes off, right? And GDP might go in and bind, but it's less likely as GTP to bind. Like what happens is you have to have a GDP that randomly diffuses, and then only then will it redock back to the GPCR, right? So you're like, instead of that single hydrolysis step, GTP to GDP, the GTP comes off and it's split into GDP. You're adding a bunch of steps to the redocking process. But regardless of that redocking process, the active GTP back state will be relatively transient, so you wouldn't get downstream signal. I think this is wrong. I'm gonna personally like block the out of the room.
Mon's gonna vindicatively grade your test.
Yeah.
Oh yeah.
Yeah.
Well sent it through.
Who got this wrong?
Who got this?
So,
All right, so in the JAK-STAT pathway, the receptors are not kinases. That's why they have the JAKs. The JAKs act as their kinase. Right, in the RTK, the receptor itself has a kinase domain. That's how the RTKs are able to phosphorylate each other. But in the JAK-STAT, they rely on JAK, which is not part of the receptor. They're not RTKs.
in an RT set, right? But in JAK-STAT, it doesn't have a kinase domain, and that's why it needs to go. Right,
It's-
remember the Jack stat is the one with the phospholactin receptor. You don't need to remember that it's phospholactin, but it's a receptor that's bound to Jack. And that Jack is between the cross phosphorylation and then the phosphorylation of the receptor. There's no autophosphorylation in Jack or in Jack stat.
Right, it's not a member of the RTK. That's why the receptors do not phosphorylate each other. Why do they have the kinase domain? On to that. No, I forgot how many questions I put in. All right, that was about the receptor. This question's about the stats.
You guys aren't getting these, that's okay. Don't worry, beat yourself up. If you only get, like Robert said, you only get a very city. So it's pretty,
I only put a minute
I
because we
see.
don't have infinite time.
that's true.
But on a test, you get like,
Infinite time.
you could take
Time.
12 minutes. You could take 20 minutes to answer this question. You could take an hour, but don't.
I remember we just talked about it. And whether or not the receptor of Jack stats has kinase ability.
We smurfed it. We got it. That's pretty good. And like on my exam from last year, there was a question very similar to this. What Laura did, she had select all that are true about Jack stats, explain why. And she just gave like 8. Like basically what I did, I just gave you 8 things. She gave us 8 prompts. You have to say whether it's true or not and explain briefly why. So this is exactly what would show up on the test. Yes? They do the receptor and the stat. Again, the receptor has no kinase domain. It does not phosphorylate anything. The receptor itself is the JAKs. And it contains an SH2 domain. That's a small difference, but it will affect you on the test, unfortunately.
That was a student in the receptor. And
All right, back to the trip operon. Oh my God, we got it. I love it. We talked about this in class today, so if you were paying attention, should be a quick answer. That's pretty good, right? Attenuation occurs because the ribosome stalls on the leader sequence. And this stalling, oh, right, it doesn't stall. Right, attenuation occurs because it does not stall on the leader sequence. And so what occurs is your ribosome is blocking region 2, hairpins 3 and 4 form. When the ribosome does stall, it gives time for regions 2 and 3 to hairpin, and that prevents the 3 and 4 hairpin, and no attenuation occurs. So it's the stalling that prevents attenuation. So if we convert it into stop codons, the ribosome will always stall at the leader sequence, and it will never attenuate. Can you read it? I really hope you have good eyesight. If you can't read it, just come up. And then I will tell you, you have to select either the yellow or the green. Right, so in this case, even though the repressor is non-functional, it does not bind to the operator. There is still attenuation that can occur. So when you have high trip levels, you're probably also going to have high trip tRNA levels, right? And so when you have those high trip tRNA levels, you're still going to get attenuation. So even if one system fails, there's a second system. That's why there's a redundancy. Right, so it's only going to be in low, medium, and what is the regulation? It's primarily through attenuation because the repressor does not work, which we looked it up. So it's going to be a very similar question, but the mutation is different. Right, so now in this example, what's mutated is your trip. What am I saying? What's the mutation is occurring in the leader sequence, so it's something to do with attenuation, right? So if we have alanine instead of trip codons and we assume alanine is always high in the cell, will the ribosome stall? Anyone? No. And so I think this question is wrong. This is wrong. Oh, all right, the question's wrong. I'm so sorry.
I think like everything is the right answer.
Yeah, there it's like, oh wow. Right, you're oh whatever.
Okay, low trip levels means low trip tRNA.
Yeah, the trip TRNA has nothing to do with this anymore because now it's controlled by our TRNA. Right? The only thing that controls it is in the context of trip versus low trip versus high trip is actually just low trip, right? And it's regulated at the repressor level because low trip is going to pull off the repressor, and so you answered that.
No, but it won't transcribe the whole transcript. You get no product because it always attenuates.
It will always attenuate, so you won't get anything.
So the question is kind of wrong. I will fix it. I'll fix it next time. What?
Shit, who went down? You like this tennis right there.
Just gonna-
Robert will give you half.
I'll give you all of it back. And I'll give you a hint. You don't have to select either the yellow or the green this time. Yeah. The entire leader sequence. All right. So what happens is if you take out the entire leader sequence, like 1, 2, 3, and 4, can attenuation occur? Okay. And then without a trip repressor, can a repression occur? No. So what is regulating the trip operon? Nothing. So it doesn't matter if it's low trip, doesn't matter if it's high trip. If there's nothing to regulate it, you'll just keep transcribing it. And yeah, there's no regulation. It's kind of easy question after that explanation.
Yeah.
That's not a true question. Oh, I should put this before. I did not think this one through. Huh? Mhm. Yeah. Okay. So, right, the leader sequence is not required for your protein stuff. That's what I'm getting at in this question. Like the leader sequence, it does not have to be functional. Even if you take out the leader sequence, all the proteins that are created are still functional. And you'll still be able to transcribe and translate tryptophan.
It's just that now attenuation would no longer control the trip operon. The leader sequence, does everyone know what the leader sequence is? Is there on that? Yeah, it's a sequence prior to the genes. It contains 1, 2, 3, and 4. And it contains the attenuation sequence. Something succeed.
This is a long question.
For any question about melon fire, you definitely will have a question about melon fire on your exam. Think about why this was happening. Like, what is the point of the siRNA pathway? Like the whole idea that doctors will do that. What is the point of that? That's very important. You will have a question about melon fire. Yeah. Robert had one. You had one in the problem set. I don't see that on.
That's pretty okay, I'll be honest. All right. So whether it uses the sense strand or the anti-sense strand, this really just comes down to like the mechanism, right? Like, how is risk detecting this mRNA? If you have a guide strand that has the same sequence as your mRNA, will they bind? Anyone? Yeah, they have to be complementary, right? So that's why the sense strand, the strand with the same sequence as the target mRNA sequence, is not used by risk. It has to be the antisense, the complementary strand, because that is what will actually bind to mRNA. And then you guys pretty much Gaia.
Go over what sense versus anti-sense.
Well, do you guys understand sense versus anti-sense? Like that the prompt makes sense. Okay. Right, like if you have duplex mRNA, one of them has to be exactly identical to the mRNA and one has to be complementary, right? And we call those sense or anti-sense. Anti means complementary, essentially.
So in the duplex, you have both the sense and an antisense. Well, melon fire put both the antisense and the sense. That's the first iteration. They have the same effect. When they added just the sense, nothing happened.
Right, so what can you infer from you had to add both? I'll give you, when you add sense and anti-sense, what happens is they couple with each other, right? They're complementary to each other, so they couple into a duplex. So it doesn't matter if you add duplex RNA or if you add sense and antisense, the sense and antisense will eventually duplex anyways.
And the other thing is if you're adding sense and antisense, that works. And if you're adding sense and that doesn't work, what is most likely doing the work? The sense and the antisense. No, it's the other way. If you drop the antisense, right? In one situation you have both, and the other situation you drop antisense, and by dropping antisense you lose function. Right?
Yeah.
Shit, never mind. Yeah. If you're okay, then never mind. So basically saying that if you're not adding both, right? If by doing that, then you lose function, then what that means is that you just need to add both.
So that's what you got. You guys got the red one. Like it has to be in the duplex form to work. And then the second thing, the blue one is a little bit less obvious. But another thing that could be happening is double-stranded RNA is just more stable than single-strand RNA. Maybe when you add just single-strand RNA, it gets degraded and it doesn't actually get to match up with risk. Yeah, it lets go of one of the strands and it keeps one of them. Now you have a question?
The duplex is made up by a sense and an antisense, right? It's like coding and non-coding, right? Like those are complementary to each other. And the same kind of thing here, where sense and antisense are complementary to each other.
I guarantee you, Laura will have a question like this on the test. You just go through the reasoning, like something about like double-strand RNA is more stable than single-stranded. Double-stranded RNA is what's actually taken up by risk, not single-stranded RNA. And then something about antisense being the guide. Like just hit those key points and you should be good for this question.
Does anyone have any other questions? Any lingering questions about anything?
So anti-trip is just another
Complementary to the coding, right? Think of it as like coding versus non-coding, right? What is the anti?
Hmm?
Is it say it's sense and anti-sense?
Coding is sense, anti-sense is non-coding.
Yeah, there you go. Coding strand, the coding strand of the DNA is the sense strand, it's the anti-sense strand.
No,
Right?
it's the sense.
What?
They're both sense is equal to
Okay,
coding.
listen to Robert.
Right. How do I describe it? When you say complementary, you have to ask yourself, what is it complementary to? Right. So your mRNA, there's mRNA in your cell that you want to target, right? Do you go about that by creating siRNA that's the exact same sequence sense? Or do you go by creating a complementary strand that's antisense?
It has to do with like the way that DNA is called coding and non-coding and also called sense and anti-sense.
Right? Like we have to figure out, you always have like two sequences and you can get one sequence from the other. We need to like identify like the characteristics of these two different sequences because they're not identical, but they are related. They're complementary.
Does anyone else have any lingering questions about anything we've gone over in the past 2 some odd hours?
It's been a long, yes.
Abby?
It's just like a quick question, because you said that phosphorylation and autophosphorylation weren't the same thing,
Yes.
but in the topic notes, it says that.
Autophosphorylation means that it phosphorylates itself, right?
But-
Cross-phosphorylation means that it phosphorylates the opposite.
It's
Right.
it's one of those things in biology where some people don't agree that they're the same thing, but other people think they're the same thing. It's one of those things that it really comes down to your definition.
You only get points off for saying one or the other, but say auto for when it's doing phosphorylating itself, literally itself, like whatever that protein is, and cross when it's doing the other in a dimer.
I mean, the perspective otherwise would be like, it's still one RTK phosphorylating itself, so it's auto.
Yeah.
Or you can think of it as like individual subunits.
That's the reason.
Okay,
Anyone
Thank
else have any other questions? Anyone online? Yes.
You know, the consensus and stuff or whatever.
Yep.
That didn't.
That might. and
thank
the
you
place
everyone
where that
for
might
coming here today.
is Whether or not.
We're done. Have a good night.
Remember that